It's really my great pleasure to introduce Professor Sara Mostafavi as our speaker for GBSBC seminar this week. Sara is currently an assistant professor in the Paul G. Allen School of Computer Science and Engineering at the University of Washington. She did her PhD in the Department of Computer Science at the University of Toronto, her postdoc research at Stanford University with Daphne Koller, and prior to joining UW, was a faculty member in the Department of Statistics and Medical Genetics at the University of British Columbia in Canada. And at UBC, she was also held a Canada Research Chair in Computational Biology and was a recipient of a Canadian Institute for Advanced Research, or CFAR, AI Chair. And Sara and her group developed a lot of really cool machine learning and statistical methods to get at the molecular basis of both cellular function and human disease. And recently, she's been tackling things that stretch from immune responses that are encoded in our genomes, psychiatric disorders, and causes of rare genetic disease in children, often through the lens of these sequence to function deep learning models. And I think we'll be hearing about several of these types of models today. So thank you, Sara, so much for joining us virtually. And whenever you're ready, please go ahead and take it away.
Thank you so much for that super kind introduction. And thank you so much for inviting me. I'm really happy to be here to tell you about some of our new research. Hopefully, I'm very excited by some of this line of new developments, and I hope I'm able to convey the reasons why we're so excited about it. But essentially, I'm going to tell you about a line of work that we call sequence-based deep learning models. Not just me, several other labs are also working in this direction. But as a promising approach to really understand the fundamentals of gene regulation and enable variant effect predictions. All right, so first I thought I would just briefly start with introducing my lab and our research in the last several years. So since I've started my own lab, I guess that's about almost 10 years ago. First I was at UBC, and then 3 years ago I moved to UW. We've been really interested in questions that focus on genome interpretation in one way or other. So we've developed models for trying to predict the impact of genetic variations across individuals on both molecular phenotypes, for example, gene expression, and also disease phenotypes. And we've developed statistical approaches for incorporating environmental effects, figuring out the context-specific effect of genetic variants, and things of that nature. But more recently, maybe 4 or 5 years ago, we kind of shifted our research to try to actually figure out how we can decode the information about cellular differentiation as encoded in regulatory genomic locations. And maybe for most of this talk today, I'm going to focus on the second direction, but near the end, you'll see how it ties in with the first direction of research that we do. Okay, so just jumping into the motivation, as you know, in our bodies, we have many different types of cells. Immune system is a nice system to study this because a lot of the different types of cells are worked out. So this is a cartoon where it's showing, maybe a handful of fully differentiated immune cells, but immunologists believe there is, 200 or so immune cell types that exist and have different functionality. And the perplexing thing is that they all contain more or less the same genome. So how does the same genome give rise to so many different cell types with so many different functions? And so the angle that we're going to look at this question is through cis gene expression regulation. And the idea being is that if you look at different cell types, for example, there's a picture of CD4 and B cells, 2 types of immune cells. What we discover is that we, as in like the field knows well, is that these cell types have differences in the regulatory regions that they use that allows them to express different kinds of genes. So in a nutshell, if we analyze regulatory regions in different cell types, we might discover what's referred to as a cis regulatory grammar that will specify, which set of genes are expressed in a given cellular context. And so it's been a long-standing question in the field that if we had all possible regulatory regions that control gene expression in a given cell type, can we then use the sequence features encoded in that region to try to figure out this cis regulatory grammar or cis regulatory logic that then specifies, which set of genes are expressed in that given context. So the good news is that, since 10 years ago, there has been huge consortium effort in identifying all possible cis regulatory elements in the human genome across many different cell types. And this is through systematic application of epigenomic assays such as ATAC-seq, ChIP-seq, cut and run, and so on, that allows investigators to identify a large collection of regulatory elements in a given sample or cellular context. So as these data through the ENCODE consortium and many others, more recently IGVF and MGEM, have been accumulating, they have allowed the creation of, I guess now what we can refer to as large sequence or DNA language models. And the idea is that if you have all these experimental measurements across the genome, we can pose a problem, a machine learning problem, that is the following. Can we train a machine learning model that takes as input genomic DNA? These could be short genomic subsequences, and predicts as output all sorts of epigenomic or regulatory features that we can measure from these subsequences across the genome. So for example, if you've applied ATAC-seq assay to measure chromatin accessibility, we can build these models to take as input genomic DNA and predict chromatin accessibility for the same subsequence across different immune cell types where we have measured chromatin accessibility. So we and many others have worked in this direction to develop what are so-called sequence to activity, or sometimes referred to as sequence to function, deep neural network model, because this model here is kind of the state-of-the-art performance is currently with really deep neural networks. And why is this interesting, or why should we care? There's two reasons why people think that this is a really interesting direction. One is that if you really believe we can train a model that just looks at genomic DNA and predicts all sorts of experimental measurement, for example, which TF bind and whether or not that piece of DNA is accessible, then maybe we can dissect the model to learn some biology. So what is the actual cis regulatory grammar that was learned by the model to enable these predictions? So that's more on, the promise of trying to understand gene regulation and biology through the lens of these large sequence models. But there's also a second motivation that's equally as appealing, which is to use these models as a sort of in silico mutagenesis engine. The idea being that once we have a trained model like this, then we can ask hypothetical questions that we can't to the extent that we could computationally, even biologically. So we could ask questions like, if I were to mutate arbitrary bases in this input sequence, how does that affect my output? And maybe this will allow us to kind of ask questions, more combinatorial questions that we couldn't ask with like perturbation assays.
Yes, Sarah, I have a question that's coming up, and I'll go ahead and ask it in the spirit of you said, hey, jump in with questions.
Yeah, sure.
In the input, do you also have gene expression?
No, I mean, you could. It really depends on how you formulate these problems, but what we refer to as sequence to function models in the input standard is just the genomic DNA. And you're right. So our predictions are going to be context specific. So if we trained a model for predicting, measurements from brain, we can predict, whether or not that piece of DNA, if there's some gene inside of it, what's the expression of that gene in the tissue for which we have training data for.
Great, thanks.
So that was a good question. Okay, and for the rest of you, please feel free to chime in. Okay, so this is the motivation. What does this talk about? I'm going to tell you about 3 projects that kind of pose questions around using and evaluating these sequence to function models. The first project, which is a little bit older work, and most of it is published, I'm going to tell you about some of our efforts in trying to actually interpret what complex models, like complex convolutional neural network models, have learned, and how do we can we learn some new biology from them. The second part, I'm going to tell you about more recent kind of systematic evaluation of how well we can actually perform in silico mutagenesis with these models. And then third project, more recent and ongoing work, trying to figure out how to improve the causal interpretation that these models have to improve our ability to predict variant effects, essentially. And this is, each project is kind of led by PhD students that I'm working with, and I'll mention them as I go through. Okay, so the motivation by the first project, we really got us started in this direction, was based on a collaboration we had with the Immunological Genome, or ImGen for short, consortium. And what ImGen is basically a consortium of about 20 immunologists that assay different aspects of immune system systematically across many known immune cell types. So in this latest data generation effort, what ImGen did was cell-sorted 90 distinct immune cell types from adult mice, which are the nodes in this dendrogram that I'm showing. So these cell types belong to like 8 broader lineages, but the point was that for each of these broader lineages, there is subtypes of cells that are also captured. And these are subtypes that immunologists, I guess, agree have some sort of differences in functionality. And you can arrange them through differentiation from hematopoietic stem cell, which would be like the white node here, and on the ends you have the fully differentiated, or the leaves you have the fully differentiated immune cell types. So the ImGen project essentially in a couple of years ago generated ATAC-seq data from all these different immune cell types. And this is another way of looking at the data. So what are we measuring with ATAC-seq? We're measuring chromatin accessibility, which we can quantify through peak calling. That's why you see pictures like this. So where do we find reads that correspond to peaks, which we can then associate with accessible regions of the DNA. So for each of these cell types, we can create these annotation tracks on top of the genome that tells us which regions were accessible according to ATAC-seq and by how much, which is kind of essentially the height of these peaks that we're getting from application of ATAC-seq. So I guess what we expect to see and what we do see is that there's a good number of regions that are accessible in all of the cell types. These are ubiquitous regions. And there are some regions that are exclusively accessible in only some of the cell types. So the initial investigation of sequence to function models in our lab started with this data set. And what we did was, in collaboration with Christophe Benoit, who's one of the leaders of the ImGen project, a postdoc in his lab, and 2 students from my lab, we created this model, which we call AI-TAC. So what is this AI-TAC model? It takes as input genomic DNA, about 200 base pairs, which are essentially regions that are found to be accessible in at least one of the cell types that was queried, and is trained using a convolutional neural network to predict as output accessibility, a continuous measure of chromatin accessibility across all the 90 different cell types. So it's a multitask model. It takes input DNA and predicts chromatin accessibility across all the cell types. And I'll give a little bit of intuition, but how does it learn that in a few slides. But the idea is that, now that we have ATAC-seq data for all these assays, we have large enough training data to actually train a model like this. And in this experiment, given the ATAC-seq in all of the cell type, there's about 500,000 OCRs, or open chromatin regions, across all the cell types, and they're about 200 base pair long. So that's what we're learning. We're learning the relationship between genomic DNA and chromatin accessibility in different cell types. So first question is that, how well can we actually make these predictions just looking at the genomic DNA? So given a genomic DNA that I haven't seen before by my model, how well can I predict which cell type will it be accessible in? So we can assess that through standard cross-validation, which I'm showing you here. So what do we measure in cross-validation? For every single input, so every single input is a sequence of 200 base pair long, I have a multitask model that makes predictions across all the cell types. So for every single input, I have a vector of length 90 with prediction of accessibility in each of the 90 cell types. So what I can do is now take that prediction vector and correlate it with the actual observed chromatin accessibility measurement. So I get one correlation value per each of the peaks or regions that was left out during my training. And if I look at the distribution of that, that's the orange distribution here. That's the distribution of the correlation values on the left out data. And I can compare that to a null, and I can construct null in different ways. One way would be a model trained on shuffled data, shuffled sequences, or I could also shuffle the cell types. And this will give me a sense of how well am I making the predictions. So we can say something like for 60% of the left out regions, the model can make a statistically significant prediction that's different from zero, essentially. And is this exciting? I said we thought it was really exciting because at this point, this is a couple years ago, we didn't realize that on just the basis of genomic DNA for a large number of accessible regions, you can actually predict the correct accessibility across different cell types. And here's an example of a particularly nice predictions. So what I'm showing you here on the top and this pink bars is the ground truth accessibility across the different cell types. On the bottom is the predictions. And we have 90 different bars, 90 different cell types. And the point that I wanted to make here is that the accessibility patterns are actually pretty complex, even within T cells, which are all very similar to each other, but there's subtypes of each other. There is variation in accessibility, and the model seems to be able to learn that variation. Okay, so yes, there's a question, Adam.
Yeah, I'm curious, kind of a couple things about the peak sets, right? Are they pretty uniform in terms of the number across cell types? And in cases where basically you maybe don't have as many peaks for a given cell type, does that make it more difficult to predict on those? And might that justify a model solely for that particular cell type instead of doing the multitask version of things?
That's a really good question. I used to have a couple of slides on that when I used to give this talk earlier, but now I added other stuff that took away that. But in terms of this data set, there are some, there is differences in number of regions that are accessible in different cells, and that certainly affects your ability to predict. Like, so if there's a cell type that looks like every other cell type, you don't have that much signal for the specificity of that cell type. And some of the T subtypes are like that. But I guess what I will say is that one thing that we played around a lot is what form of the loss function you use. You can emphasize different aspects of the data and how you learn those. So the specific loss function that we used here was to actually optimize predictions for signal that is exclusive for a given cell type, and not so much the signal that's shared, because we wanted to understand the differences. And I guess that's the reason why I like multitask learning in general, because then you can compare the different predictions in a coherent way, versus if you had a single model per cell type, it's hard to compare models because there's also the overall training that makes the models different. But I mean, I guess that's an ongoing research question, and there's pros and cons in different ways of doing it.
Makes sense. Thanks.
Okay, so I guess with this, we spend a couple of years to convince ourselves that this model actually learns something interesting. And some of the other results that are in our paper, and I'm not showing, for example, we showed that you can train a model on mouse and make sensible predictions on human, and all sorts of other kind of experiment that really convinced us that there's something that the model has learned that might be interesting. So then that made us foray into this emerging area of so-called explainable AI algorithms. And the idea is that neural networks and complex models in general have historically been viewed as black boxes. So people didn't really care to understand why they make certain predictions. It's just a really large model that can kind of predict outputs from inputs. But now that they're being used in science and biology, and more generally, there's lots of interest in trying to figure out how do they actually learn. So foraying into this direction, and looking at many different algorithms, we actually ended up writing a review article more geared towards genomic audience. For this, talking about different ways one can go about interpreting what trained models have learned. But let me tell you about the simplest strategy that we used that was effective. And the idea is very simple, and it was kind of 2 steps of it were borrowed from some previous papers. But let's go through it. So first of all, how do, if we train a model, what is the principle for how it learns to make predictions from genomic DNA, in particular convolutional neural networks? So how they learn is that they take as input, use it based on one-hot encoding of the input sequence. And then we have different hidden layers in the neural network that learn different subsequences and their combination that are predictive for the outcome. And in particular, the nodes in the first hidden layer, each one is learning to recognize a short sequence motif that we can represent using kind of the standard position weight matrix or PWM logo. So these are position weight matrices that might correspond to kind of regulatory elements such as or regulatory units, I should say, such as transcription factor binding motifs and other kind of regulatory binding motifs. And so once these shorter subsequences are learned, they're being combined together in the deeper layers to make the predictions. So the approach that we use, it has 3 steps. So in the first step, what we do is we, for every hidden node in the first layer, we can actually directly either look at the weights or look at its activated sequences to reconstruct the position weight matrix that was learned that hidden unit is trying to detect. So for each node here, we have one kind of PWM that the node is detecting. And just the way our architecture worked is that we had 300 of these hidden units in the first layer. So we're going to have 300 of these PWMs. That's our first step. The second step is we're going to determine the importance of that PWM in models prediction. And the way we do this is through ablation. So basically for each hidden node, one at a time, we're going to nullify it, set it output to 0, and then measure how the predictions of the model change after we nullified it to figure out how important it was for making predictions. So if we nullify one of these PWMs and we realize that now the model is making a lot of errors, that is a signal that says that was a really important PWM that was learned. So through this second step, we compute some sort of motif influence. And the third step, which was kind of what we added to these 2 previous steps that were pretty standard, was to have some assessment of computational replication or reproducibility. And the idea is that because these models are basically the parameters that end up being learned depend on the initialization, what we need to know, what we need to do is also address uncertainty in the parameters that are learned. So the simplest way to do this is through ensembling, where basically we train a number of different models on the same data and repeat step 1 and 2 to identify motifs and compute their significance or influence. And this allows us to have some sort of computational replication value associated with each motif or PWM that was learned. So let me just show you a few quick results on this portion of the talk. So what did we actually see when we tried to interpret this model? This plot, what I'm showing you here, is that each dot here is one PWM that was learned by the model. On the x-axis, we have the information content of the PWM. So how different is it from just random ACTGs? And on the y-axis, we have the influence of that PWM on the model's predictions. And the colors are the most important part here. So they're color-coded by reproducibility metric that I just explained. And so what we saw here was that basically, and this now it's very obvious, at the time it was also speculated, that models like neural networks are over-parameterized. So they learn many features that might not be necessary and maybe just represent the noise in the training data. And that's essentially what a lot of the filters or a lot of the motifs that are learned representing. They're not reproducible across models runs. But there are some portion, approximately about half of the motifs or PWMs that were learned that seems to be reproducible. But a good number of them are also not. So it's an important metric to pay attention to when we want to interpret what neural network models like this have learned. There is a question in Q&A. Okay, so the question was, and Alex, please clarify if I didn't understand correctly, but the question is how many features were fed into the model in using one-hot encoding. So in this case, I mean, they're not proper features, but yes, we call the input features. The features are the base pairs. So the number of base pairs in a given sequence would be equivalent to the number of features. And in this case, our model is trained on sequences that are of length 200 base pair long. So in that terminology, you can assume that we have 200 features that are one-hot encoded. Okay, so I think that was a good answer to Alex. So the second plot that I wanted to show was, so did we learn, is there any biological validity to what the model learns? Because the model can make good prediction, but it doesn't have to be biologically coherent. It could be based on some rules that doesn't make sense to us. So this was a really exciting observation for us, that the model actually tends to learn, rediscover biology that we know. So how do we show this, or how do we actually investigate this? One thing that we can do is we have these PWMs that are learned, and we think that the way, biologically, the way this would work is that these PWMs should represent transcription factor binding motifs that are present in accessible regions. And that's how the model is figuring out where a given region is accessible. So to investigate this, what we did was, for each motif that was discovered, we matched it up using kind of alignment algorithms with known transcription factor motif databases such as SysBP and JASPAR. So now what I'm showing you here is each dot is one PWM that's learned. We have the information content and influence on the X and the Y axis, but now the colors are the things to look at. So the color indicates that if there was a match for that PWM that was learned in a transcription factor database. And I guess this was super exciting because it said that, we are learning fundamental biological regulatory mechanisms by just using this ATAC-seq data and training a complex model like this. We're recovering a lot of the known transcription factors that are known to be important for immune cell differentiation. And finally, the last figure that I'll show on this part is that because we have this multitask model, so going back to Adam's question, what we can do is ask basically each of these. So if I go back to this picture here, where we're ablating each of these PWMs and looking at how much influence it has in predicting our output, what we can do more precisely is look at the influence of each of these PWM across each of the cell types that we're predicting. So we can create these heat maps like that for each PWM that we've learned. Now we have the transcription factor annotation, and we can look at the influence of that learned PWM across the different immune cell types, which are the columns here. And again, we can validate this in different ways. So for example, we can actually align the predictions with known RNA-seq gene expression. So for example, there's this PAX5 transcription factor that's known to be really important for B cells. We can look at the learned influence of PAX5 according to the PAX5 learned PWM, and contrast that with the actual expression of PAX5 across immune cells. So each dot here is a cell, and then we see really nice alignment. So I guess this was a really good data set for us to evaluate how well models learn, because so much is known about the immune system, which transcription factors are important. and things like that. So now I'm going to move on to the second part of the talk, which is how well can we use these models to actually perform in silico mutagenesis analysis. But maybe I'll pause to see if there's other questions on the first part. Okay, so, but feel free to ask questions if it comes up later. So I told you about one model that we developed, but there's tons of these models now being developed for predicting different kinds of epigenomic features or gene expression from different kinds of cellular contexts. But there's one thing that almost all of these models have in common, which is actually hard to realize if you don't work in this domain. And the thing that they have in common, why do they work, is that they basically all formulated a machine learning problem that uses genomic regions as examples. So these models are all trained on single reference genome, essentially, but you have so many training examples because there's so many regions where you have measurements for. So in our case, where we're looking at accessible regions, there's hundreds of thousands of accessible regions, and each region constitutes one data point. There's many other models, the state of the art being Informer, that was in Nature Methods just last year. What they did was kind of the same training recipe, but using larger input sequences. So instead of 200 base pair, around 100 kb input sequences to predict gene expression and other kind of epigenomic features. All these models have been shown to actually perform really well in a certain type of evaluation. And that certain type of evaluation, which I have a picture of on the right, is if you leave out regions, for example, you leave out regions on a chromosome, you can predict the experimental measurement on those left out regions really well. But the main kind of like one key promise of these models is that once they work well, we can use them to perform in silico mutagenesis as following. So the idea is that once we have a trained model, we can give it the reference sequence, which maybe it's like something like on the top sequence I have here, and then introduce an arbitrary mutation from a T to a C and look to see how the predictions of the model change in any kind of measurements that the model was trained for. So for example, if it's a model to predict gene expression, then you can see introducing a C here, how does it impact my gene expression prediction? And we can do this systematically for every base in a given genomic sequence. We can even do it combinatorially for every pair of bases in a given genomic location. And there's been some promise of why this should work, even though, as I mentioned, these models have never seen genetic variation data, right? They've only seen genomic sequences in a single genome, and they've learned to predict experimental measurements. But there's some promise that they can actually do this variant effect predictions because, and the logic is like, if you've learned the cis-regulatory grammar of gene regulation, then you understand if you change this T to a C, maybe you modify the transcription factor binding site, and that is going to have an important consequence. So there's
Sarah,
been, yeah.
I have a question, I think. Oh, actually, I think they accidentally asked maybe me only. I can read it out though.
Sure.
Daniel, he asks, how do you evaluate the role of epigenetics in the whole story? You probably have no way to normalize epigenetic marks in your experiments. Therefore, is there a way to know what is more important between epigenetic marks? Like examples are histone or methylation versus the DNA sequence specificity in driving the expressions of genes.
Yeah, so that's the question. I mean, you're studying that in an indirect way, and there is models for specifically addressing that question. I guess the models that I'm describing are models for explicitly understanding what happens if you change DNA, what happens to gene expression or other measurements. And those models are cell type specific. So you can say for a particular, in brain cells, if I change this base, it will impact gene expression in this way. But what you're not in this way that I formulated modeling is that the joint effect of genetic DNA and, let's say, DNA methylation at a locus. So if that joint effect, like maybe methylation, basically overrides the genetic variation in that region and impacts gene expression. So that's not what you're modeling, although you could model it that way. The data is just not as clean for creating those large models for answering that question compared to what we're trying to do here. But there are offshoots of methods that try to explicitly answer that.
Awesome. I think that answered it.
All right. So yeah, there has been like some promise in that this will work. And even though these models aren't just trained on like a single reference genome, you can make good predictions from them. So for example, in the Informer paper, what they did was you can do these like in silico mutagenesis, which is this top track shown here. And you can actually line that up with MPRAs and show that it's pretty consistent in select loci with these empirical, with MPRA experiments. So that could be pretty revolutionary if you actually have an in silico version of MPRA, right? But I guess it's like kind of, it was still debated that how generalizable these results are. So we thought we would do it in a different way. We would evaluate these models in a different way in a recent paper that just put out in bioRxiv last year, and it's going to be in Nature Genetics in a few weeks. But the idea is that we can actually use natural genetic variation in large whole genome sequencing projects across individual to evaluate the prediction of these models, which were not done in the past. So the idea is that, we have a model that takes as input and takes as input genomic DNA and predicts as output, let's say, expression values. Well, if you have multiple individuals, and in particular multiple personal genomes, what we can do is take as input these personal genomes across different individuals and get the predictions of the model for each of these personal genomes for a given locus, and look to see how well the model actually makes this prediction. So this was also a joint collaboration with Maria Chiquina, who's at University of Pittsburgh, and a couple of our students. Okay, so so this is the evaluation we did. We had access to the ROSMap project, which is a large multi-omic project that has WGS or whole genome sequencing data paired with RNA-seq for about 800 individuals. And let me first tell you about one locus that actually the model like Informer works really well, and then tell you about systematic evaluation overall. And I should mention that Informer is not our model, but we know that this phenomena that I'm explaining is a very generic phenomena, and we've validated, and there's now other papers that describe it for other sequence to function models. So I'm not targeting Informer, it's a general principle. So let me first tell you about just this one gene, make predictions for this one gene. So there's one gene, DDX11, and it's a positive example of when things work greatly. It's really great. So this gene is easy because from just statistical fine mapping studies, we know it has a single causal variant that impacts this gene expression across the population. And also we know that the gene expression of that gene is really highly heritable, which means that basically that one genetic variant can explain gene expression variation in a population. So what we're going to do is, in this gene, DDX11, there's about 400 variants in the population of the samples that we have. We're going to insert these variants into the reference genome to construct, personalized genomic DNA centered at this gene DDX11 and look at the results. So that's essentially what we do. We have 800 or so personal genomic subsequences centered at the transcription start site of DDX11. And the length of these input sequences is about 100 kb, which is the input window for Informer. And feed those into Informer and get predictions for gene expression from the cage track. There's a cage brain track that actually matches the data that we have. And this is a nice example where you can see the observed gene expression across the individual. So in this scatter plot here, each dot is an individual. And you can see the predicted value by Informer. And it's highly correlated. And you see these, I guess, 3 categories of individuals. And that makes total sense because there is a single causal variant. And depending on the number of minor alleles of that one you have lower or higher expression of the gene. So this was a nice example. And the reason why it's nice is that Informer or models like it have never seen genetic variation, but somehow they've learned the logic of gene regulation that now looking at these personalized genomic sequences, they can make accurate predictions. And then we can further go and figure out why did they make that prediction, so we can do in silico mutagenesis around the known causal variant, which we didn't tell the model. And what we observe is that, so this is our plots that you would get in silico mutagenesis analysis, where the height of the base indicates the importance of that base in the model's prediction. So essentially what happened in this locus was that there was a reference T that has reduced expression compared to the allele that has the A. And why that happens is that the A results in a repressive motif. So if you have the A, you have lower expression, and A results in a repressive motif. So you can basically go and figure out why the causal variant impacts gene expression if you have a model like this. There was a question, will you consider expanding this model to include long read RNA-seq coupled with AI tag? Maybe that was from, maybe I can get back to that question when near the end. Okay, so that was a result of like one positive example of things working well. But what about the general case? So in the case of DDX11, what I showed you here is like for each gene, basically we can make predictions across the individuals and correlate that with the observed to get one correlation value. So the one correlation value for this gene is 0.85, but where the correlation values look more systematically across all genes. So that's shown on the y-axis, each dot is a gene, and the x-axis is the population average gene expression in the cortex, which is where the predictions and the data is coming from. So what you see, I guess the surprising thing that first jumped out is that there is a substantial set of genes that the model predicts the wrong direction of the effect. And that's why you see a negative Pearson correlation value. What does that look like? So here's an example of a gene GSTM3, where basically the model predicts that the variant should decrease the expression, but the actual reality is the opposite of that. The variant increases the expression. That's why you see this negative correlation between observed and predicted. And it turns out that that's pretty commonplace. So one observation is that the ability to predict variant direction is not very calibrated. The other observation was that now we can go and figure out how well actually we're predicting gene expression by comparing models' prediction with kind of standard statistical genetics approaches for quantifying gene expression heritability from cis variants. The idea being that gene expression variation in the population has some genetic component, but also has some other trends or environmental component that's not captured just by sequence and by genetic information. And we can quantify for a given gene how much of the gene expression variability is explained by genetic variation through basically association analysis. There's an approach called PredictScan that does it pretty well. So now what we can do is compare Informer's prediction with PredictScan, and what we see, and let me just summarize for the purpose of the time here. But what we do see is that basically Informer is nowhere near having the ability to really predict gene expression variation correctly. So there's a bunch of genes that the direction of effect is predicted incorrectly. And even these are for genes where we know most of the variation is explained by genetics. Okay, so why is this happening? And this brings us to the third segment of the talk. So by analyzing these models and really trying to understand when they make mistake, I guess our current hypothesis is that this happens because models like Informer or standard sequence to function models really don't have enough training data to learn all subtle biochemical sequence features that can influence gene expression or other functional measurements. So this is an example of motivating the hypothesis that I just said. For most of the loci, if you look at the ISM values in that loci and look where the known causal variants are, the known causal variants are not falling into well delineated motifs that are learned by the model. So all of that kind of motivates us to hypothesize that there's just more training data is needed. Even just looking at subregions in the reference sequence is not enough to really learn the consequence of genetic variation that generalizes. So that brings us to the third project. So how do we do this? And that's work done by current PhD student Shiming. The idea is that let's go back to the mouse data, which is simpler to debug and study. So the same way that we can do in silico mutagenesis on Informer and other models that I mentioned, we can also do it in our setting where we trained data on mice. So there's also different strains of mice. All of the model that I described in the first part of the talk was trained using data from B6 mice, which is kind of like the reference mice. But we can also do the same test where we train a model on B6 mice and ask how well does it predict, for example, chromatin accessibility in one of the cell types that was measured in a different strain, Cas strain. So Cas and B6 have about 20 million variants between them. And we see the same thing, that the model is not really good at figuring out the impact of genetic variation. So the correlation and prediction decreases a lot once we go from B6 to Cas. So this gave us an idea of basically we need to use natural genetic variation as some sort of perturbation to enhance what these models learn. And to do this, we collaborated with Christophe Benoit to generate F1 hybrid mice. So what is the significance of these mice? So I mentioned that we have the B6 strain, we have the Cas strain. We can create these hybrid strains. And these hybrid strains are special because the parent strains are each homozygous across all their genome because they're inbred. But once we make these F1 hybrid crosses, they're basically heterozygous across most of their genome. So why is that interesting? So now if we generate something like ATAC-seq data or RNA-seq data would have the same property, we can separately map reads to the maternal and the paternal haplotypes, or the B6 versus the Cas haplotypes. And we basically are faced with a really well-controlled perturbation experiment across the genome. So we can have three types of cases where we have the Cas strain and the B6 strain, and we can compare the effect of genetic variation on chromatin accessibility. So we can have a case like this where here's the Cas allele, here's the B6 allele for a given region. Maybe there is a variant inside one of the alleles, but the chromatin accessibility is the same. So this is an example of where genetic variation doesn't impact chromatin accessibility. But we can also have a case like these two, where we have our B6 and Cas allele, and we have a genetic variant, and that genetic variant could either increase accessibility or decrease accessibility. So if we look across the genome for all regions that are pulled down by ATAC-seq, in general, we expect majority of the regions to be allelically balanced, even if there is genetic variation. So they have the same accessibility in both alleles. So this is the kind of the ratio between the reads from the two alleles in a given region. But there's a subset of regions that actually are allelic imbalanced. So how do we use this data? So essentially, we have a bunch of paired alleles that provides really clean training data for models. And these paired alleles are in a very well-controlled experiment because they're both living in the same cell, and the accessibility difference should mainly have a genetic explanation. So to utilize this data, we propose this model deep allele, that what it does is that it takes as input, so it's like a contrastive model. It takes as input pairs of alleles in a given region, so one allele from B6 and one allele from Cas, and predicts as output the allelic count and chromatin accessibility from each of the alleles, as well as their log ratio. So in this way, once we train the model, we can hopefully answer the question of impact of genetic variants a little bit better, because there is more training data for the model to learn the effect of genetic perturbation on chromatin accessibility. And how well does it work? So we can compare the prediction of difference in the allelic accessibility in unseen region, which is closer to asking more directly what's the effect of genetic variation on kind of the output measurements. And here's 4 different data sets that we've used to train and test the model on. So there was 2 different ATAC-seq data sets and 2 different ChIP-seq data set, all from F1 mice. And what we can see is that the deep allele does better than our original model AI-TAC in predicting the effect of allelic difference. And this is also better, the model deep allele is also better than a very simple way to use all the data, which we call multi-genome. So essentially train the same model AI-TAC, but on all the genome, except don't tell the model this pairing allele information. Okay, so I just have, I think, 2 more slides, and then we can get to questions if there are any. So we observed that it works, and now we want to figure out, why does it work better? What did it learn that the vanilla model didn't learn? So what we can do is look at regions where our model deep allele made better predictions and try to figure out what are the features learned in those regions that are not seen in the reference-based model. So what do we do? So here's like the prediction of our model DA and prediction of the baseline model that just uses this reference genome called single head model. And we can identify regions where our model is not making a mistake, but the other model is making a mistake. And I guess this will be a little bit anecdotal because this is still ongoing work, but I thought it's still very interesting to share. So what do we see is that usually in these regions where our model does better, turns out that the model learns sequence context a lot better than a reference-based model does. So here's an example of performing in silico mutagenesis in one region that our model does a lot better than AI-TAC. And if you look at what's different, basically, so our model finds 3 motifs. So there's one motif here, one motif here, and one motif here. And the variant, there's 2 variants that occur in the secondary motif. And our model realizes that this secondary motif is important for prediction of accessibility in this entire region, versus the reference-based model, AI-TAC, doesn't learn that secondary motif, presumably because it was not tied to genetic perturbation data. So here's a second example where this is an example of a region that the top plot is from our model and the second plot is from the reference-based model. And what's happening in this region is that it's a region that contains many copies of Pu1 binding sites. Oh, sorry, and I should have mentioned, this is actually ChIP-seq data. It's Pu1 ChIP-seq data. It's not the ATAC-seq data. So there's this region that contains many copies of the Pu1 binding site that, it's just present in the sequence. But the difference is that our model downweighs the contribution of these multiple Pu1 sites when they're all occurring in a row, and understands that genetic variation in one of them is not going to be so impactful because there's this buffering effect, versus the reference-based model doesn't understand this buffering effect. So that's all I had. So hopefully you're excited as I am about these application of these sequence-based models and discovering biology from them. So in the first part, I told you about our model AI-TAC for trying to understand gene regulation in immune cells. In the second part, I told you about systematic evaluation of in silico mutagenesis, pointing to the fact that there is still ways to go to get these models to work well. And in the third step, I told you about kind of our current approach to try to address some of the shortcomings of the model identified in the second part. And these are all the people that I've collaborated with on the projects. Christophe Benoit is a good collaborator at Harvard. Phil Diager and David Bennett on the model evaluation using paired genome sequencing and RNA-seq data. And these are all the students that have contributed to these. And sorry for going fast, but happy to take questions.
All right. Awesome. Thank you, Sara, for the wonderful talk. Are there any questions from the audience? Feel free to put it in the chat. I'm happy to read it out or raise your hand and ask. If not, I'm also happy to kick things off, but I already asked one, so I'll let others. Farhat, do you have a question you want to ask?
Yeah, it's in the QA. I don't know if you guys are seeing it at all.
Oh, I see it. Do you want to?
I can put it in the chat. I wasn't sure. I mean, it's a long question, but I can make it very short. So this is all great work, and I love these sequence to function models. But when you're evaluating them, I think when you're saying mistakes or the effect size, it's predicting in the wrong direction. So we need to keep in mind the LD structure of the EQTLs and SNPs and so on and so forth, that the validation data or the genetic correlations that you're finding, some of them are likely not correct themselves, right? The validation data is not correct, that it is not a causal SNP, it is in LD, sometimes in the opposite direction with a causal SNP and so on and so forth. So it might be that the model is not really model may say there's no effect, and then you see this beautiful effect, but the causal allele of that beautiful effect is not that SNP. So model is actually right, not wrong, right? I mean.
Oh, right. Okay, so we've thought a lot about these questions, and I think this is a common misunderstanding if you're coming from statistical genetics. So the way these models are trained is that they have no notion of LD, and I don't know if there's a picture that shows that, but maybe there's no picture necessary. They have no notion of LD because they've only been trained on a single reference genome, so there's no LD structure to learn, right? So what they're learning is if there is some sort of sequence grammar that is important for like predicting gene expression, right? So when we apply them, actually what we see is that in cases where they work, and maybe this is a good example to show, like DDX11 is one case that it works. It actually doesn't identify a bunch of variants in LD, and I don't know if I can show it from here. Right, so this is like in silico mutagenesis in the DDX11 locus, where we know there's one causal variant. If you were using EQTLs, you would see a bunch of other variants here. But what this model does is like in one shot identifies the causal variant because there's no notion of LD here. And we're not evaluating with EQTLs because you're right, there is LD structure. Like EQTL doesn't mean it's causal. You have to do statistical fine mapping. But we're asking a slightly different question, which is more subtle, which is given a complete genotype, can you correctly predict gene expression? And there's no confounding by LD in that question.
So that your plots where you showed that the effect size, this plot especially, I thought here you are looking at the Pearson correlation, the anti-correlations, positive, negative. This is with respect to your predictions versus the population variation of gene expression with respect to that SNP, is it?
No, it's not per one SNP. It's the entire genotype at a given locus and using that as input to predict gene expression.
Okay, so you are taking really large chunks of DNA, like the whole genome sequencing, the entire. How big of a region that you can handle here?
It's about 190 kb.
Okay, so most LD structure you're going to be able to capture. Okay, great. So I misunderstood. Thank you.
Thank you for that question, Joe. I mean, I think we also were really confused by this fact, but it's a subtle but really important insight that there's no notion of LD in sequence-based models unless you train them on multiple genomes.
Yeah, so that was why I was saying if you don't have any notion there, if you have it in the validation data, then but you don't have it. That's great. Thank you.
Yeah, I guess I missed these in the Q&A instead of the chat. There was one that you read out. I don't know if you want to try to take a stab at long read RNA plus AI attack.
Let me go back to that question. Right. Will you consider expanding the model to include long read RNA-seq coupled with AI attack? I think it would be a really interesting direction. Definitely worth investigating. We haven't gone into that direction, but yeah, I think there's like lots of interesting directions that could come out of it. And then
I think the next one would be from Monica. And then also it's at 101. So feel free whenever you need to go.
all right, maybe I'll just take the question from Monica. What is the lowest number of individual genomes used to train the model that you haven't seen to still result in accurate predictions? Okay, so I guess that relates to the third part of the talk. So what we're doing now is actually training these models on multiple whole genome sequencing data from multiple individuals. And it's a hard question to answer because we don't know yet, but I guess for cases for easy genes, let's put it, there's some genes that are very heritable and the regulatory structure is very easy. You probably need about 30 to 40 individuals to really understand the relationship between sequence and gene expression. But there's other genes that are a lot more complex and probably you need in the order of hundreds of individuals to try to predict them well. But one, I guess, so maybe this is the last point that I make is like the main difference with doing an S2E model or S2F model like this versus population-based association analysis is that with these models, your sample size is actually multiplied by number of genomic regions. So you actually have a lot of data to learn from, and that's purely because of the way the model is formulated.
Cool. Awesome. So I think if you have other questions, I feel like you probably could feel free to email Sara. Thank you so much for joining us today. This was a really awesome talk and some really cool stuff you got going on.
Thank you so much. It was really a pleasure. And thank you for all the questions.
Yeah, thank you, Sara, and thank you, Adam, for hosting.
Yeah, absolutely. Hopefully next time we'll see you in La Jolla.
Awesome. Have a good day, everyone.
Great. Bye.
Thanks, everybody.